In this episode of our series, we'll look at why classification is important, the Linnaean system, how molecular biology led to new systems, and evolutionary trees. Let's explore the principles behind biological classification. Classification is fundamental to how humans process information, from organizing libraries to structuring databases. In biology, this organizational instinct becomes a vital scientific tool. By grouping the sheer number of organisms on Earth based on shared characteristics, scientists can identify patterns and understand relationships between different forms of life. Without classification, biological research would lack the structure needed for meaningful analysis. The modern classification system originated with Carl Linnaeus, an 18th century Swedish scientist. He developed the Linnaean system, which arranges organisms into nested groups, each level more specific than the last. This hierarchical structure creates a logical framework for understanding biological diversity. The classification hierarchy consists of seven main levels: kingdom, phylum, class, order, family, genus, and species. Each level represents a different degree of relatedness between organisms. This system functions like a postal address, beginning with broad categories and progressively narrowing down. Just as an address moves from country to specific street number, classification moves from kingdom to individual species. For over 2 centuries, this framework provided the foundation for biological classification. Traditionally, scientists recognized five kingdoms: animals, plants, fungi, protists, and bacteria. This division was based primarily on visible characteristics and basic cellular structure. However, advances in microscopy and molecular biology revealed that traditional classification based on physical appearance didn't accurately reflect evolutionary relationships. Scientists discovered that organisms appearing similar externally could be very different at the molecular level. In 1990, Carl Woese proposed a revolutionary three-domain system based on genetic analysis, which transformed our understanding of life's most basic organization. Through RNA sequence analysis made possible by new technology, Woese identified three fundamental divisions of life that supersede the kingdom level. The three domains are archaea, bacteria, and eukaryota. These represent the earliest evolutionary branches in the tree of life. Archaea are single-celled prokaryotes often found in extreme environments, such as thermal vents and salt lakes. Despite superficial similarities to bacteria, they possess unique biochemical characteristics. These organisms demonstrate remarkable adaptations to conditions lethal to most other life forms. The bacteria domain encompasses true bacteria, including familiar species like E. coli and Staphylococcus. Though prokaryotic like archaea, bacteria have distinct molecular and biochemical properties. Eukaryotes include all organisms with nucleated cells and membrane-bound organelles. This domain contains the kingdoms of fungi, plants, animals, and protists. This classification reveals surprising relationships. Humans share more fundamental characteristics with fungi than bacteria share with archaea. Such insights demonstrate the value of molecular classification. Within these three domains, the traditional Linnaean hierarchy remains useful. The system continues to provide detailed classification below the domain level. Scientific naming follows strict international conventions to ensure clarity. The binomial nomenclature system assigns each species a two-part Latin name. The first part indicates the genus, while the second identifies the specific species. For example, humans are designated Homo sapiens, Homo denoting our genus, and sapiens our species. This systematic approach eliminates vagueness in scientific communication. Binomial nomenclature provides a universal language for scientists worldwide. Regardless of native language or location, researchers use identical names for the same organisms. Evolutionary trees visually represent relationships between species based on common ancestry. These tools help scientists understand how different organisms evolved from shared ancestors. Branch points on these trees indicate common ancestors, with closer branches suggesting more recent divergence. The structure reveals evolutionary relationships that may not be apparent from physical characteristics alone. Scientists construct these trees using multiple data sources, including DNA sequences, protein structures, and fossil evidence. Modern computational methods allow analysis of vast amounts of genetic data. This comprehensive approach provides insights into evolutionary history spanning billions of years. By integrating various evidence types, scientists can trace relationships from microorganisms to complex multicellular life. Next time, we'll examine biodiversity and explore why maintaining species variety is critical for ecosystem stability.